We put a ton of trust in our doctors, right? And we trust the tests they use to figure out what's wrong with us. But what if one of those tests, a test for a really deadly disease, tells a flat-out lie? So let's just jump right in. Picture this, a test. One that doctors count on for life or death calls is just wrong. What happens then? What happens when the data itself is the thing that's deceiving you? All right, so let's set the scene. You're in a hospital ER. A little kid is rushed in really sick. We're talking fever, stiff neck, all the classic, absolutely terrifying signs of bacterial meningitis. So the team, they jump into action, right? They do a spinal tap to get a sample of cerebral spinal fluid, or CSF, and they rush it to the lab for the standard test that's supposed to give them the answer. And this is where it gets weird. This is where our mystery really starts. The lab calls back, and the results? Totally normal. The fluid's clear, no sign of infection whatsoever. But the kid is right there in front of them, and they are obviously dangerously sick. You've got the test telling one story and the patient telling a completely different one. What do you do? Okay, so to even begin to unravel this, we gotta understand the place where these tests are actually done. We need to take a look inside the microbiology lab. So the first thing you need to know, the absolute #1 rule in a microlab, is something called aseptic technique. The easiest way to think about it is like a sterile bubble. It's all about two things: keeping random outside germs out of the patient's sample, because that would give you a wrong answer, a false positive, and keeping the potentially nasty germs in the sample from getting out and making someone sick. It is, no exaggeration, the foundation of everything they do. And here are the two main tools of the trade. You've got the microscope, which lets technicians literally see these invisible little bacteria, and then you have the culture plate. That's where they actually grow the bacteria to figure out exactly what they're dealing with. So it's kind of this two-step dance. First, you spot the invisible culprit, and then you give it a place to grow so it becomes visible to everyone. Yeah, that culture plate, it's a lot like a little garden. A microbiologist takes a tiny bit of the patient's sample and kind of plants it on this special gel that's packed with nutrients. And then they wait. They're basically watching to see if any dangerous weeds, the bad bacteria, start to grow in their little garden. Okay, so now that we know how the lab works, let's circle back to our big mystery. When they're testing for meningitis, what exactly are they looking for in that spinal fluid? All right, in a textbook case of bacterial meningitis, the lab expects to find three things, three big clues. First, a ton of white blood cells. That's your body's army showing up to the fight. Second, they should be able to literally see the bacteria under the microscope using a special dye. And third, when they do that gardening thing and put the fluid on a culture plate, those bad bacteria should start growing like crazy. And check this out. This is what that growth actually looks like. It's so cool. Different bacteria make these unique little patterns called colonies. You can see they have different shapes, different sizes, some even have different colors. These visual clues are everything. They're how the lab figures out the exact identity of the bad guy. So that just brings us right back to the million-dollar question, doesn't it? If this kid really has meningitis, how could all three of those signs be missing? No army of white blood cells, no bacteria to see, nothing growing in the garden. How on earth could that test come back completely normal? Well, the answer, believe it or not, was buried in a medical study from all the way back in 1990. A study that found a super rare but incredibly important exception to the rule. So what happened was, a group of researchers looked back at old records from two different children's hospitals. They pulled up the files for 379 kids who were all confirmed to have had bacterial meningitis. And their goal was simple. They just wanted to see, hey, how often did that first big test actually get it right? And as they sifted through all that data, all 379 cases, they found something small, a tiny little anomaly, but it was a number that would change everything. That number was 4. Just 4 cases where the first test was dead wrong. So let's break down those 4 cases. In 2 of them, the test results were completely, totally normal. Sound familiar? Just like our kid in the ER. The other 2 had just a tiny abnormality, something you could easily miss. But here's the kicker, the piece that connects it all. In 3 of those 4 cases, the kid had been sick for less than a day. And because of those normal tests, the study points out that getting them the right medicine was significantly delayed. That's a huge deal. And that is the answer to the whole puzzle. The infection was just too new. It was in the very, very early stages. So yeah, the bacteria were there, but there just weren't enough of them yet. And, even more importantly, the child's own immune system hadn't had time to sound the alarm and launch a full-scale attack. Without that massive response, you don't get the huge rush of white blood cells. And without that, the infection is basically invisible to our standard tests. So this whole story really shines a light on a massive challenge that doctors face all the time. It's what we could call the clinician's dilemma. It's this fundamental clash, right? On one side, you've got the test results, the hard data, the numbers on a screen that say, hey, this patient is fine. But on the other side, you've got your own two eyes, your gut feeling, your years of experience as a doctor screaming that the person in front of you is not fine, that they are really, really sick. So which one do you trust? And this is what it all comes down to. The authors of that 1990 study wrapped it up with this incredibly powerful warning, and it's really the main takeaway. They said that a normal test result does not and cannot rule out meningitis. A doctor's suspicion, what they see with their own eyes, has to be what guides them. So here's the bottom line. Lab tests, they're incredible. They're powerful, essential tools that give us information we couldn't get any other way. But that's what they are. Tools. They're one piece of a much bigger puzzle. They're there to help a doctor make a decision, not to make the decision for them. That human element, that art of medicine, is still absolutely vital. And that really leaves us with a big question to chew on, doesn't it? We live in an age of data. We want the numbers, the algorithms, the objective facts to guide us in everything. But this whole story is a pretty stark reminder, you know? It makes you wonder, when it really counts, when the stakes are literally life and death, how much do we trust our own eyes? How much do we trust our gut?